for the kind introduction. I'm Ranjan Batra. Can you hear me now? Okay, it's better. Great. So I'm based off of San Diego, and I work for this company called Locana Bio. And I'll give you a little bit of history for that. And if there's any questions after the presentation, you can always email me. It's my first name, last name at locanabio.com, Ranjan Batra at locanabio.com. So what is Locana? It has a relatively short history. It was founded in 2016. So one of my great friends and colleagues now, he had a great discovery in the lab. We were working for Genio, our postdoc mentor, and he found that he can use a CRISPR molecule, and I explained what that is in a little bit. And he found that instead of targeting DNA, he could target RNA. And at that point, I had worked on DM1 for about 10 years. So I started my PhD in Maurice Swanson's lab in 2007, and I completed in 2013, worked for a year at Mayo Clinic, and I was in UCSD in Genio's lab at that point. So I had this idea that if I can use Dave's system to target RNA, in fact, CUG-repeat toxic RNA, then maybe we could actually treat DM. So I used his designs with Gene's guidance, put it into the cell models, and lo and behold, it actually worked. So that was great. In the time that we were about to submit our work for publication, I moved up to San Francisco for a year. And I came back when the company was funded. It was founded by Dave Nellis and Genio, who couldn't be here today. So in 2017, we got licenses from UCSD and started our work, which is something that I'm going to present in the next few slides. And recently, we were selected as the top candidate for some non-dilutive funding from the MDA for venture philanthropy. So what is Locana platform? What do we do? So we use this protein called CRISPR protein. And this is the number 2 on your list. So I'll just explain briefly what our platform consists of. Oops, sorry. So CRISPR protein comes from bacteria. Bacteria have defense systems against viruses. And viruses have DNA and RNA that they put in the bacteria. And the bacteria defend themselves by producing this protein called CRISPR that goes and binds to viral RNAs and DNA, so these are foreign toxic RNAs, and degrade them. So they defend themselves with that. Earlier in this decade, there was a lot of work done at UC Berkeley and Broad, and they showed that these systems can be adapted in humans. So we actually used this system to target a different foreign RNA, a different toxic RNA that was the DM CUG-repeat RNA. So this system consists of a guide RNA molecule. It is, a guide RNA gives this system specificity, so we can target specifically the CUG RNA and not any other RNA, so that it's safe. And then it has a protein that helps this guide RNA to bind to the target. And then we fused this system with an enzyme that can actually destroy the RNA, the toxic RNA. So essentially, we have a search and destroy system. We search for the toxic RNA, that is DM1 or DM2 RNA, and then basically destroy it. So this was our hypothesis that in DM1, there's CTG repeat expansion. They form these toxic hairpin loops that sequester or bind these proteins like MBNL proteins. And essentially, most of the phenotypes are manifested due to the loss of function of these muscle-blind proteins because they're so tightly bound by these toxic RNAs. So our hypothesis was that if we use our system to bind and degrade this RNA, we can release the muscle-blind protein to do its normal function. So we took our system, we packaged it into a gene therapy vector, which we call AAV9, shown in the middle. And we injected mouse models of myotonic dystrophy. These are HSALR mice produced by Charles Thornton. And we injected the tibialis anterior muscle, which is the shin muscle. And we used this model in such a way that in the left leg, we injected with a control that does not express our system. In the right leg, we expressed our system. So essentially, what we want to see that if we give this drug in the right leg, should be cured, and the left leg should be the same. And first of all, we followed the expression of the system for 3 weeks, 3 months, 12 weeks. And we saw that the expression of the system in muscle is safe as well as durable. And this bar plot just shows you that even at 12 weeks in different dosage regimen, the protein is expressed. So anything above the dotted line shows you that the protein is expressed. Protein and the RNA for our system are expressed at good levels, at therapeutic levels. And this continues to happen for another few weeks. That data I'm not showing you today. So when we used this system in vivo or in mice, we wanted to see the reversal of the disease, or DM. First of all, we wanted to see if the foci that are formed by CUG repeats or toxic CUG repeats get reverted or get eliminated. And we saw the same thing. On the left, it's the left leg that was injected by a control, not targeting the CUG repeats. On the right, we targeted the CUG repeats using our system, and the foci were eliminated in the mice. This is just a quantification of the same result, and we see it works a little bit better in this case in females just because they have a smaller muscle mass. But when we increase the dose, males are also, it also works in males very well. So lastly, we wanted to actually get an up-close look to see if it works, basically a high-resolution look, if it works. And sorry, it's stuck. Oops. I guess the image is not showing, but it basically shows that, oh, it is showing now. So the green fibers are the ones, the green muscle fibers are the ones that got our system. And the dark ones are the ones that did not get our system because we're expressing a proxy protein that's green in color. So all the blue dots are nuclei where the toxic RNA should be. And you can see that all the fibers that are green do not contain foci anymore. The CUG RNA was gone from the foci. So it was really therapeutic in mice, in the muscle fibers that showed expression of our system, or that got our system. So this made us very, very excited, because now we have a system that we can put in mice, and we can eliminate CUG foci, CCUG foci for DM2, and so many other microsatellite expansion, repeat expansion diseases. Saying that, translation of biologics to humans is really hard. So we have a system that works in mice. We are very excited. We want to take it to the next level. But to get to humans, we have to tackle the problem of delivery. So we're actually doing a lot of work in non-human primate models, as well as some other big animal models to see that we can translate this. But again, it's an exciting time because there are many players in the field. Although translation is hard, there are more shots on the goal. So it makes me excited that something is just around the corner. So saying that, I think this video will really show you how the drug works. And this is going to show you functional improvement in myotonic dystrophy in these mouse models. And in these mouse models, we basically injected them intravenously. So this is a separate injection or experiment from the one that I showed you. And we're looking at myotonia in these mice. So I'm going to play the video now. So this video was produced by Dave Nellis, who's my colleague. So we injected these mice intravenously at birth. And we're looking at 8 weeks after. And we assessed myotonia by this pulse pull test. So when we pull a little bit on the hind limb, the muscle gets stuck, and it's unable to relax because it's myotonic. But when we do that in treated mice, which is showing you now, the myotonia is gone. So this is at 50% speed side by side. So you're very happy to see that we had cured this myotonia or myotonic disease in this mouse. Saying that, our tentative timeline is unsure as of now because we're working on our financial plan as well as talking to investors, as well as talking to some regulatory agencies to see when we can actually take this to the clinic or start talking to the FDA, and we will keep you updated. And I just wanted to say that we have a great team, and we're really committed to building a gene therapy for myotonic dystrophy. And like I said, our CEO is Jinyo, and he could not be here. He was here for some other conferences. Or he was here for the MDF conference last time, but he couldn't make it this time. And on the behalf of Locana and everyone in the team, I wanted to thank you for listening intently. And I also wanted to thank our SAB members and academic collaborators. And I'll take any questions. Thank you.